All right, let's talk about measuring populations. And I'm going to try not to shove my face with goldfish as we go through this together. It's going to be so much fun. So fun, so amazing, so great. Okay, so there's 5 main agents of change here. Mutation, gene flow, non-random mating, genetic drift, and selection. We'll talk more about each one as we kind of go through the Hardy-Weinberg. So just know that these are things that bring about change. Remember, evolution is change over time. So these are the things that are going to bring about that change. Mutation, obviously a change in DNA. Gene flow is when you have like an actual individuals moving, emigrating or immigrating to a population. Non-random mating, when you have more controlled mating. That would be the opposite of that. Genetic drift, we've talked about the founder's effect, the bottleneck, and then selection. Predators, in this case, that very hungry bird, picking the worms it can see. So we have predator selecting for certain traits for its snacks. Okay, here we go. Population and gene pool. So here we go. Population is just a localized group of interbreeding individuals. You cannot see that color, so I'm going to change it. The gene pool, way better, is a collection of alleles in the population. Remember, alleles are different from genes, okay? You've got one gene, essentially, is eye color, hair color. It's very general. For every gene, you're going to have 2 alleles, okay? You're dominant and you're recessive. Allele frequency is how common that allele is, right? How many dominant and recessive alleles are in an entire population. All right, so populations change. Again, change, evolution is all about changes. What? You heard me. And we're talking about allele frequencies, how common these alleles are. How common is that dominant allele? How common is that recessive allele? So the Hardy-Weinberg is very hypothetical, right? It's like, what conditions would cause allele frequencies to not change, to stay the same? So that's what you have. You have a non-evolving population, right? A population that does not see any of this change, kind of remains constant, very predictable, right? So you get rid of all these things, like we just saw 2 slides ago. You have a large population size, right? So you've got no genetic drift. No migration, no one's moving, no mutation, no random mating, no natural selection, right? Nothing. So if you remove these, it kind of gives you this theoretical situation. And the theoretical acts as a model for us to compare other populations to kind of help us better understand. It acts almost like a control group, or, when we do a controlled experiment. Here we go. Hello, Hardy-Weinberg. Hardy and Weinberg. German's most eligible. So it's hypothetical. Every population evolves. We know that. So the fact that there'd be a non-evolving population just doesn't exist. But that's okay, because it gives us something to compare it to. And it preserves this allele frequency, okay? And it serves as a model. That's the job. That's its goal. And hey, talk about models, am I right? Check out those handsome guys. You always know when you're going to see a German thinker by the dome situation here. Okay. Natural populations are rarely in Hardy-Weinberg equilibrium, like I said. It's a model. It gives us something to measure actual populations by for us to better understand them. So what is this all about? It's about alleles. You've got 2 alleles. Here are our cats. Meow. You've got 2 alleles, right? So say we're looking at fur color. Again, one gene, fur color, going to have 2 options, black or white. All right? P, we know, is the dominant allele. Q is the recessive allele. They must equal 100%, so we know the formula is P plus Q equals 1. Those are alleles. Those are this business. Now, if you're looking at something different individuals, say you want to get into the actual genotypes. So when we talk about individuals, we talk about genotypes. You're going to have to use this equation, okay? So the frequency of this genotype would be p squared, 2pq, and q squared. All right, different, because we're talking about alleles. I'm sorry, we're talking about individuals, genotypes, that business. Alleles, easy peasy, p plus q equals 1. Individuals or genotypes, it's going to get a little bit more complicated. Here are oh my God, did I repeat the formulas? So here are the formulas in case you forgot them when I said them 10 seconds ago. Here we go. Alleles, individuals. Individuals, you can also remember genotypes, right? That's what I'm talking about. So let's give an example of this. You have a population, you have my dream population, right, of 100 cats. Say I go to that awesome island in like Southeast Asia where all they have is cats, and because I finally like lose my mind, you guys eat all the goldfish and I freak out, and then I just say forget it, and I get in my Volkswagen Jetta. You heard me correctly, and I drive to an airport and I go to the island, and then a boat, I guess. I'd have to get on a plane and a boat. Anyway, I end up in my paradise, also Cat Island. And I see 100 cats, and 84 are black, 16 are white. So we want to know how many of each genotype. Well, we should always start with the recessive, because we know that genotype. If there's a white kitty cat, we know it's this genotype, always. So if there's 16 white, 16 divided by 100 is 0.16, okay? That's our q squared. Great, we know that. Once we know our q squared, we can then find our p squared, and our 2pq. So our q squared is 0.16, okay? We get that 16 from this number. There's 16 kitty cats. There's out of 100, so the frequencies for q squared is going to be 0.16. Okay, so say we take the square root of this to get q, and we get 0.4. Then our p is going to be 0.6 because this needs to equal 1. Great, now we can do this. Now we know 48 is 2 p, which is 6, times q, which is that gives you 48, and p squared is just 0.6 squared. Okay, and that's it. And that's all we need to know. So when we talk about, I'll talk more about the null hypothesis. So this is kind of like the ideal, right? If you have 100 cats and you've got this, these are going to be your genotypes. But say you get cats, you do a lot of research on them, and you get, this is your actual, right? Okay, well, there's a difference here, right? There's a difference here. So then you can start to look at, well, what's happening here? Why is there more heterozygotes than we thought? Is there possibly a heterozygote advantage? Okay, is it more advantageous? Is it more, does that make the organism more fit? Okay, if there's more heterozygous, if you're heterozygous as opposed to homozygous recessive. So let's talk about an example of this, and this is kind of this idea of being a fit heterozygous, okay? And you have your sickle cell anemia, right? This is actually your sickle cell. It's shaped like a sickle, right? So you have this mutation, which we saw when we talked about protein synthesis, for hemoglobin, all right? So this protein in your blood that carries oxygen is jacked. So now you have your red blood cells look like this. They can't carry oxygen as effectively as these guys can. And you've got problems. You don't have oxygen going to your whole body. You have a lot of people who are in pain. You have people who are having mini strokes because they're not getting oxygen to their brain. All of that horrific stuff, okay? So this is your recessive. This is if you had the sickle cell. And this is kind of like your normal allele. I don't know why they put an S here and a B here, but just go with it, okay? So if you've got low oxygen levels, that causes your red blood cells to sickle, which is what this looks like, and it's damaged, and it's often lethal because you're getting, what happens is people a lot of times will get strokes, and that's kind of, or irreversible damage to organs. All right, so let's talk about Central Africa, shall we? So you have a high frequency of heterozygotes. Again, if you have 100 people in Central Africa, you should only have, what, 48% of the population based on the Hardy-Weinberg, right? That's the ideal. And I got that number, say from, I kind of made that number up. So you get this number, it should be about 50-ish% in a perfect population. So the problem is here is if you are not heterozygous, if you're homozygous, if you're homozygous recessive, you have sickle cell. If you're homozygous dominant, you have a likelihood of contracting malaria and dying. So there's an advantage here. So you're going to see a higher rate of people surviving in Central Africa who are heterozygous. So they compared that population to the ideal, this Hardy-Weinberg, and that's where this comes from, this idea of like, oh, wait a minute, there's more heterozygotes here. Why? Well, it's better. There's selectivity there. Malaria, right? Malaria is the disease. It's caused actually by a parasite. It gets in your blood. Ugh, frowny face. It damages the red blood cells. It replicates like crazy. It's just not really great. To say the least. Boom, boom, beep, bop, beep. So here you go. So you have this. You're going to get malaria. See ya, wouldn't want to be ya. This, you're getting sickle cell. Bam. Heyo. This, you're going to have an advantage. You're going to be more likely to survive in Central Africa. Why am I telling you this? Because, again, this is a way that we use the Hardy-Weinberg to point out these anomalies in populations. Once we can find the anomaly, we can say, okay, what's happening here to cause this certain genotype to be more favorable? That's basically it. I hope I made sense. Now, if you excuse me, I'm going to go stick my face in a bag of goldfish. Peace out.